Hi everyone, welcome to my presentation on small airway specific epithelial cell biomarker expression in normal and cystic fibrotic lungs. My name is Esonica Charles and my mentors are Dr. Kenichi Okuda, Hiraki Murano, and Dr. Jim Haggard. So to introduce, cystic fibrosis is a genetic disease that causes thick mucus to build up in specifically the lungs, since this is the organ that I'm working with. And usually when there's a lot of lung buildup, a lot of mucus buildup in the small airways, this leads to obstruction and a decrease in lung function. So currently, recent single cell transcriptome analysis has identified different epithelial cell types in large versus smaller airways. However, it is unknown how small airway-specific epithelial features are altered in cystic fibrotic small airways. The objective of my study is closely related to HuBMAP's overall goal, and the HuBMAP goal is to develop a platform to map healthy cells in the human body for different organs and organ systems. So we wanted to identify localization of small airway epithelial cell specific biomarkers, which includes SCGB3A2 and SSFTPB in normal and CF lungs, and then compare the expression levels between normal and CF small airways. The next objective was to identify correlations of expression levels for total SSFTPB and SCGB3A2 with the upstream transcription factor NKX 2.1. And then we wanted to test if small airway epithelial cell cultures retain those specific biomarkers, which can be distinguished from large airway cell cultures. So to do this, we dissected tissue from 10 normal and 10 CF donors at transplantation under the approved UNC IRB protocol. And then we did immunohistochemistry for those samples. Hiraki helped me a lot with doing this for my project, and I learned a lot of the basic immunohistochemistry protocol. And then to quantify our data, we use QPAT, which is an imaging software. And here on the right, this is what an image would look after it's been scanned through a slide scanner. And then here's how QPAT recognizes the DAB positive cells for our calculations. Finally, for in vitro large and small airway epithelial cell cultures, we're used to measure transcript of small airway markers in each region. Here I have my results of some images. So I have a general image of a normal lung tissue. And here we have large going for NKX 2.1, and then for the biomarkers ProSFTBB and SCGB3A2. And here we have the DAB expression shown in like the darker purple areas. And then for the small airway epithelial airways, we see even more of the dark purple expression of the live DAB positive cells. Then we move to CF, which all of these are DAB staining, done with DAB staining. And here we see that there is a reduced expression of the small airway markers in the CF large airways and for CF small airways. So moving on to my figures. So here we have normal versus CF, DAB positive areas in the transcription factor NKX2.1 for SFTPB and SCG, SCGB3A2. So on my y-axis, I have the percentage, and on my x-axis, I have the normal versus CF. For NKX 2.1, there was a significant difference between the expression of the DAB positive cells in normal versus CF. And here we have a much more diverse population for pro-SF-TPB. And the reason why we think that it is not as significant as NKX 2.1 is because of, one, we only tested one airway for all of these markers as of right now, and two, we think maybe it could be like the number. We might need to increase our number of samples. And then for SCGB3A2, we have a significant difference. Although it's not as expressive as NKX 2.1, it's still significant, even with those outliers that you see here in the individual colors of green and purple. So moving on to the correlation graph, we have pro-SF-TPB on the y-axis and NKX 2.1 on the x-axis. And here we see some positive correlation between the expression of total pro-SF-TPB and NKX 2.1. And the same thing is here with SCGB3A2, that there is some positive correlation between the upstream transcription factor NKX 2.1 and with the biomarkers pro-SF-TPB and SCGB3A2. So moving on to some in vitro data, we have mostly on this graph here, for small airways, we see that there is a high expression for SSF-TPB and for SCGB3A2 noted by the p-values. And it was just nice to know that the data that we did somewhat correlates to previous in vitro data that was done by the lab. So to conclude, SCGB3A2 and pro-SF-TB expression was specific to small airway epithelia in both normal and CF airways, which is positively correlated with NKX 2.1 expression. Based off of my figures, my representative figures, the expression level of these small airways were significantly reduced in CF airways when compared to normal, and in vitro small airway epithelial cell cultures express those markers. So in the future, we would like to systematically identify region-specific molecular features within the human lung, and we would do this by utilizing multiple modalities. Then we would integrate those region-specific molecular features with function, and then identify how these region-specific molecular functions may be altered in diseases. And then once we identify those, we could possibly look for treatment in clinical settings. So I would like to thank the University of North Carolina at Chapel Hill for giving me this opportunity to learn more about the lungs and to get into this type of research, which I had a lot of fun doing. I would also like to thank the HuBMAP program and the NIH. And I would again like to individually thank my mentors again, Dr. Kenichi Okuda, Hiraki Murano, and Dr. Jim Haga. Thank you.